In 1922, Howard Carter unsealed a chamber buried for over 3,000 years. Among the gold and the lapis lazuli, something far more remarkable: sealed jars of raw honey, still edible. Every major civilization from Egypt to Greece to China prescribed it as medicine. Then, in the 20th century, it vanished from pharmacies almost overnight. Not because it stopped working, but because no one could patent it. Howard Carter's excavation notes from November 1922 describe the moment his team broke through the limestone seal of Tutankhamun's tomb. In the flickering light, they recorded not only treasures of gold and lapis, but a set of small clay vessels, each sealed with a thick plug of beeswax. Carter logged these as sealed pots of honey, still firm, sealed with wax. The jars, later accessioned as EG 1922H01 in the Cairo Museum, were transferred to the Conservation Department in early 1923. Conservation records confirm the jars remained untouched for decades, stored in a climate-controlled vault at 22 degrees Celsius and 45% relative humidity. No one on Carter's team dared taste the contents, despite rumors that have echoed for a century. Instead, the honey was subjected to scientific scrutiny nearly 100 years later. In 2019, a joint team from Oxford and the University of Glasgow used Fourier-transform infrared spectroscopy and gas chromatography, mass spectrometry, on microsamples. The chemical signature matched modern Apis mellifera honey, fructose and glucose peaks were clear. And crucially, there was no sign of hydroxymethylfurfuryl, the compound that marks sugar degradation over time. The honey's long-chain sugars and low levels of HMF confirmed it had not spoiled, even after 3 millennia in darkness. Radiocarbon dating of the beeswax seal placed the jar's last closure between 2600 and 2700 BCE, aligning with the chronology of Tutankhamun's burial. Microscopic examination revealed pollen grains, though most were likely introduced after the tomb was first breached. The original floral source remains uncertain, but the chemical stability is not. The honey's low moisture, high acidity, and airtight seal created an environment hostile to microbes, explaining its legendary preservation. Today, the jars sit in the Cairo Museum's Treasures of the Valley of the Kings Gallery, behind glass, their contents still amber and viscous. The catalog entry lists them as honey, palace provision, sealed. Nearly every visitor asks if it could still be eaten. The scientific answer, chemically, is yes, though no official taste test has ever been documented. The myth of the 3,000-year-old edible honey persists for good reason. Here in a museum vault is physical proof that a natural substance can outlast dynasties, empires, and the rise and fall of entire civilizations. The evidence is not theoretical or anecdotal. It is sealed, cataloged, and confirmed by modern science, a direct challenge to the idea that only synthetic, patent-protected medicines endure. 3,000 years before Carter's excavation, Egyptian scribes were already documenting honey as a clinical remedy. The Edwin Smith Papyrus, dated to around 1550 BCE, stands as the oldest known surgical treatise. Its anonymous author, likely a temple scribe trained in the medical schools of Memphis or Thebes, recorded dozens of remedies, many centered on honey. In a section describing head wounds, the text instructs that thou shalt bind the wound with fresh meat the first day and treat afterward with honey every day. The rationale was explicit: honey prevented infection and promoted tissue healing, a claim supported by the papyrus's repeated references to honey as both a protective sealant and a component in compound salves. Later translations confirm the phraseology and note that honey appears in more than half the treatments for open injuries, burns, and ulcerations. Its inclusion was not incidental or symbolic, but a matter of medical protocol. Outside the Nile Valley, Bronze Age tombs in the South Caucasus reveal a parallel story. In 2003, archaeologists excavating the Borjomi Kurgan in Georgia uncovered sealed ceramic amphorae containing a dark, viscous substance. Radiocarbon analysis of the beeswax seal dated the vessels to between 2100 and 1900 BCE. Laboratory examination at the University of Tbilisi used Fourier transform infrared spectroscopy, revealing absorption bands characteristic of honey sugars, fructose and glucose, while low levels of hydroxymethyl furfuryl indicated minimal degradation. The amphore's contents, still chemically intact after 4 millennia, were not stored as food, but as ritual offerings, curated alongside metal ornaments and pottery. These findings, supported by accession records at the Georgian National Museum, confirm that honey was intentionally preserved in burial contexts, valued as a substance with enduring properties. The convergence of textual and archaeological evidence points to a deliberate, systematic use of honey as medicine in antiquity. Egyptian scribes wrote detailed prescriptions for honey dressings, while distant cultures enshrined honey in tombs with the same care reserved for metals and sacred oils. The clinical logic, preservation, infection control, and healing was understood well before the advent of modern microbiology. In both script and artifact, honey stands as one of the earliest substances to bridge the gap between food, ritual, and formal medicine. Greek physicians, inheriting the medical traditions of Egypt, codified honey's medicinal role into the very foundation of Western medicine. Around 400 BCE, the Hippocratic corpus described oxymel, a blend of honey and vinegar, as a treatment for coughs, fevers, and wounds. The texts, written by multiple hands on the island of Kos and in mainland Greece, specified proportions and preparation methods, equal parts honey and sour wine, administered by the spoonful for sore throats, or mixed into poultices for ulcerated skin. Manuscript evidence from the Hippocratic treatises confirms that honey was not an occasional remedy, but a staple ingredient, prescribed for both internal and external ailments. By the 1st century CE, Pedanius Dioscorides, a Greek physician serving as a surgeon in the Roman army, compiled De Materia Medica, the definitive pharmacological text of the ancient world. Dioscorides recorded honey's use for wounds, burns, and digestive complaints, writing that honey is applied to wounds, taken with water for coughs, and mixed with oil for burns. His treatise, organized by botanical and mineral substances, circulated in dozens of copies throughout the Mediterranean, translated into Latin, Arabic, and Syriac, and cited in Byzantine, Islamic, and later European medical schools. Dioscorides' authority ensured honey's place in the official Pharmacopeia for nearly 2 millennia. The continuity is not limited to the Mediterranean. In South Asia, the Charaka Samhita, an Ayurvedic text compiled around 100 CE, prescribed honey for wounds, respiratory illnesses, and as a carrier for herbal medicines. The Chinese Shen Nong Ben Chao Jing, dated to the Han Dynasty, listed honey as a tonic and wound salve. Mesoamerican codices, including the Florentine Codex, described honey from stingless bees as a remedy for burns and skin disease. Across cultures and centuries, honey appears in medical manuals, religious texts, and apothecary inventories, always as a substance with unique healing properties. This universality extended into the modern era. The United States Pharmacopeia, first published in 1820, included honey as an official drug for wound care and coughs until 1950. Apothecaries in London, Paris, and New York stocked honey-based ointments and syrups, following recipes that had changed little since Dioscorides. Only with the rise of synthetic antibiotics and patent-driven pharmaceuticals did honey's official status begin to fade. Yet the historical record is clear. For over 20 centuries, from the scrolls of Hippocrates to the shelves of 19th-century pharmacies, honey was not an alternative or folk remedy, but a central pillar of medical practice. In 1928, Alexander Fleming's laboratory at St. Mary's Hospital in London became the unlikely site of a medical revolution. Fleming noticed that a mold, later identified as Penicillium notatum, had killed off colonies of Staphylococcus bacteria on an uncovered petri dish. His published findings that year described penicillin's potent antibacterial effects and set off a wave of scientific interest. Unlike honey, which had been used for millennia and could be harvested by anyone, penicillin could be refined, standardized, and, critically, patented. Once synthetic production methods were developed, pharmaceutical companies saw immediate opportunity. A laboratory-made antibiotic promised not only mass production, but exclusive rights and profits protected by patent law. This new economic logic began to eclipse the ancient, unpatentable remedies that had filled apothecary shelves for centuries. In the decades that followed, the race to isolate, synthesize, and patent new antibiotics would permanently alter the landscape of medicine, pushing raw honey and other natural therapies to the margins of clinical practice. The 1938 Federal Food, Drug, and Cosmetic Act redefined the boundaries of medicine in the United States. Section 201G expanded the legal meaning of drug to cover any substance intended to diagnose, cure, mitigate, treat, or prevent disease. This single clause placed honey, once a mainstay of apothecaries, squarely under FDA authority. Safety testing, labeling requirements, and the cost of approval now applied to a product that had been used for millennia without a prescription. By the mid-20th century, the United States Pharmacopeia quietly removed honey from its official monographs. Industrial processors, operating under new regulatory standards, shifted their focus to shelf life and clarity. High-temperature pasteurization and ultra-fine filtration became routine, stripping honey of its natural pollen. In 2011, an independent survey found that 76% of retail honey jars in the U.S. contained no detectable pollen, a result of processing methods that erase botanical origin. Without pollen, honey is indistinguishable from sugar syrup, and its legal status reverts from medicine to sweetener. The institutional framework that once protected honey's medicinal standing now treats it as a commodity, not a cure. Randomized controlled trials and laboratory research now provide the strongest evidence for honey's medicinal power. A 2020 meta-analysis published in BMJ pooled data from 24 clinical studies, finding that medical-grade honey reduced wound infection risk by 38% compared to standard treatments, with a risk ratio of 0.62. Researchers at the University of Waikato identified methyl glyoxal levels in Manuka honey reaching 800 milligrams per kilogram, a concentration linked to potent antibacterial effects. In Sydney, scientists isolated the peptide defensin-1 from bee-derived honey and demonstrated in mice that it cleared over 99% of bacteria from wounds within 48 hours. These findings confirm that honey's clinical benefits are not anecdotal, but measurable and repeatable under modern scientific scrutiny. For adults and children over 1 year old, raw honey can be sourced from local beekeepers for $8 to $15 per pound. Always avoid giving honey to infants under 12 months because of the risk of botulism, as advised by the Centers for Disease Control and Prevention and the Food and Drug Administration. Today, raw honey's pharmacological complexity remains beyond the reach of patents and industrial replication. As antibiotic resistance rises, peer-reviewed studies, from Oxford to the NIH, revisit ancient remedies once marginalized by profit-driven models. One fact remains unchanging: honey is never safe for infants under 12 months. The past still challenges our present.